This certificate of analysis highlights the key quality attributes confirmed at batch release. The assay result lands at 99.8%, well within the 95 to 105% specification. Impurities remain tightly controlled, with each individual impurity below 0.10% and total impurities at 0.14% against a 0.50% limit. Dissolution performance meets expectations, with 98% release at 30 minutes, and moisture content is low at 0.45%. These data represent a point-in-time snapshot of product quality generated under controlled conditions at 20 degrees Celsius and verified following 92 days of accelerated aging at 40.1 degrees and 76.2% relative humidity. Pharmaceutical quality relies on two complementary pillars, the Certificate of Analysis and the Stability Program. The COA is the promise. It confirms that the batch meets all predefined specifications at the moment of release, much like a clinical checkup. The Stability Program is the proof. It monitors long-term performance to ensure the product maintains quality throughout its shelf life. These functions are governed by distinct regulatory frameworks. COAs align with EU GMP Parts 1 and 2 in FDA Q7A Section 11.4, while stability programs follow 21 CFR 211.166 in ICH Q1A R2. A scientifically sound COA must provide clear product identification, including the name, batch number, and full manufacturer details for traceability. It must specify both the release date and the expiry or retest date. Most importantly, it must present actual numerical results alongside acceptance criteria rather than relying on generic complies statements. Each COA requires a dated signature from authorized quality personnel who operate independently from production. While this document certifies current compliance, it does not predict future stability. That responsibility lies with the Stability Program. The credibility of a COA depends on the underlying GMP controls. Manufacturing is performed by qualified personnel who follow documented training in hygiene standards. Facilities and equipment undergo routine calibration, validated cleaning, and contamination control. Comprehensive documentation systems maintain both master and executed batch records. Above all, an independent quality unit, organizationally separate from production, holds exclusive authority for batch release. These safeguards, outlined in FDA Q7A, ensure the COA reflects true product quality rather than procedural shortcuts. Stability programs serve four core scientific purposes. They establish shelf life by evaluating degradation kinetics, define appropriate storage conditions for labeling, identify degradation pathways in vulnerable quality attributes, and confirm that manufacturing changes do not compromise product longevity. These activities are mandated under 21 CFR 211.166, which requires written protocols designed to assess stability throughout the intended shelf life. To support worldwide distribution, stability studies must reflect ICH climatic zones. Long-term studies for temperate regions use 25 degrees Celsius and 60% relative humidity, while hot and humid zones require 30 degrees with 65 or 75% humidity. Accelerated studies at 40 degrees and 75% humidity for 6 months provide global comparability. Certain products require specialized conditions, such as refrigerated storage at 5 plus or minus 3 or frozen storage at minus 20 plus or minus 5. These conditions ensure product integrity across diverse environments. Stress testing deliberately degrades the active ingredient, typically by 5 to 20%, using heat above 50 degrees, acidic or basic hydrolysis, oxidative challenges, or photostability exposure. The goal is to demonstrate that analytical methods are truly stability indicating. Chromatographic profiles must show clear baseline separation between the API and its degradants, confirming the method can detect and quantify impurities throughout shelf life. This approach aligns with ICH Q1A R2 and Q2 R1 expectations. Bracketing and matrixing designs improve stability program efficiency while maintaining scientific rigor. Bracketing evaluates only the extremes, such as the lowest and highest strengths or container sizes, based on the assumption that intermediate configurations behave predictably. Matrixing distributes testing across time points, assessing representative subsets of batches while preserving statistical validity. Both strategies require justification in regulatory acceptance, and they are especially valuable for product portfolios with multiple strengths or packaging variations where full testing of every combination would be impractical.
Stability commitments evolve as products progress through development. Early clinical phases rely on short-term data from technical, often non-GMP batches, just enough to support the dosing duration. Registration requires full ICH Q1A compliance studies on at least 3 primary production scale batches, including 12 months of long-term data and 6 months of accelerated data. After approval, annual surveillance testing of commercial batches confirms ongoing process consistency. This phased approach balances patient safety with development efficiency, ensuring appropriate stability coverage at each stage without over-investing in extended studies before the formulation and process are finalized. Shelf life is established through statistical modeling aligned with ICH Q1E. Data from at least 3 registration batches are pooled to characterize degradation kinetics. The expiration date is set at the point where the 95% one-sided confidence interval intersects the specification limit, not the average trend line. This conservative method accounts for variability and analytical uncertainty. Extrapolation beyond observed data may be justified when accelerated studies show no meaningful change and degradation pathways remain consistent. The resulting shelf life provides strong statistical assurance that future batches will remain within specification throughout the labeled period. Differentiating out of specification from out of trend results is essential for proactive quality oversight. OOS results represent true specification failures and require full GMT investigations. OOT results, by contrast, signal emerging process drift, such as slowly rising impurities that remain within limits but deviate from historical behavior. Monitoring these trends provides an early warning system, enabling corrective action before patient risk develops. When used effectively, trend analysis transforms stability data from a compliance requirement into a powerful tool for continuous quality improvement. Change control safeguards stability when manufacturing conditions evolve. Adjustments such as new packaging suppliers, site transfers, or formulation refinements trigger risk assessments followed by bridging studies. These studies compare pre-change and post-change materials under identical stability conditions to confirm equivalence. A practical example is moisture-sensitive capsules that failed at 18 months in polyethylene bottles. Switching to foil-laminated closures cut moisture uptake half and extended shelf life to 36 months. This evidence-based approach ensures that changes strengthen rather than compromise long-term product quality. Biologics require specialized stability strategies due to their structural complexity. They're vulnerable to aggregation, deamidation, and oxidation under conditions that small molecules tolerate. In-use stability becomes especially important for products requiring reconstitution or multi-dose use, where sterility, potency, and particulate control must be maintained throughout the labeled in-use period. These expectations for biologics reflect this complexity through ICH Q5C and product-specific guidance. Maintaining supply chain integrity requires continuous COA traceability from manufacturer to patient. Authentic certificates must accompany each batch through transport and distribution. When repackers or distributors modify primary packaging, they assume full responsibility for stability, including generating new data to confirm performance in the updated container closure system. Shipping studies verify that products withstand transport conditions, but gaps can arise when brokers intervene between manufacturers and end users. Preserving this chain of custody ensures that the quality verified at release remains valid through final administration. Together, the Certificate of Analysis and the Stability Program form a unified quality system. The COA confirms present compliance, the promise of quality at release. The Stability Program provides future assurance, the proof that quality endures through shelf life. This continuum spans the entire journey from batch release to patient use. Statistical tools such as regression modeling and trend analysis convert raw data into predictive insights that prevent failures and protect patients.